Right now, as you're watching this video, something absolutely mind-blowing is happening inside your head. Billions of electrical signals are racing through your brain at speeds up to 268 miles per hour. Your neurons are firing, synapses are connecting, and an intricate network, more complex than any supercomputer ever built, is working overtime. Just so you can understand these words, here's what's crazy. Your nervous system contains roughly 86 billion neurons. Each one of these neurons can connect to up to 10,000 other neurons. Do the math, and you're looking at potentially 100 trillion connections happening inside your skull. That's more connections than there are stars in the Milky Way galaxy. And yet, most people have no idea how any of this actually works. Think about this for a second. Every thought you've ever had, every memory you've made, every movement you've ever taken, the feel of a cold drink on a hot day, the pain when you stub your toe, the butterflies in your stomach when you see someone you love. All of it comes down to this one system. The nervous system isn't just important, it is quite literally who you are. So, today, we're going deep. We're talking about everything from how a single nerve cell fires, to why you pull your hand away from a hot stove before you even feel the pain. We're covering the brain, the spinal cord, the peripheral nerves, the autonomic system, neurotransmitters, reflexes, sensory reception, and so much more. By the end of this video, you're going to understand your nervous system better than most medical students after their first semester. And I promise you, once you understand how this system works, you'll never look at yourself the same way again. Let's start with the basics. The nervous system is essentially your body's communication network. Its job is to receive information, process that information, and then respond to it. Sounds simple, right? But the way it does this is anything but simple. Your nervous system is divided into two main parts. First, you've got the central nervous system, or CNS. This includes your brain and your spinal cord. Think of it as command central, the place where all the major decisions get made. Then you've got the peripheral nervous system, or PNS. This is everything else. All the nerves that branch out from your brain and spinal cord, reaching every corner of your body from the tips of your fingers to the ends of your toes. The peripheral nervous system itself breaks down further. You've got the somatic nervous system, which controls voluntary movements. When you decide to pick up a cup of coffee, that's your somatic nervous system at work. Then there's the autonomic nervous system, which handles all the stuff you don't have to think about. Your heart beating, your lungs breathing, your stomach digesting, all automatic. And the autonomic system splits again into the sympathetic nervous system, your fight or flight response, and the parasympathetic nervous system, your rest and digest mode. But before we dive into all these divisions, we need to understand the basic building block of the entire system, the neuron. Neurons are the cells that make everything happen. They're the reason you can think, feel, move, and experience the world. And while there are about 86 billion of them in your brain alone, each one follows the same basic blueprint. A neuron has three main parts. First, you've got the cell body, also called the soma. This is where the nucleus lives, containing all the genetic information. It's the neuron's control center. Coming off the cell body, you've got these tree-like branches called dendrites. The word dendrite actually comes from the Greek word for tree, and when you look at them under a microscope, you'll understand why. These dendrites receive signals from other neurons. Think of them as the neuron's antennae, constantly picking up incoming messages. Then there's the axon. This is a long thin fiber that extends from the cell body and carries signals away to other neurons, muscles, or glands. Some axons are incredibly short, just a fraction of a millimeter. Others can stretch over 3 feet long, running from your spinal cord all the way down to your toes. At the end of the axon, you've got axon terminals, sometimes called synaptic terminals or terminal buttons. These are the endpoints where the neuron communicates with the next cell in line. Now here's where it gets really interesting. The way neurons communicate is through a combination of electrical and chemical signals. Inside a single neuron, the signal is electrical. But when that signal needs to jump from one neuron to the next, it becomes chemical. This process is what makes everything in your nervous system possible. And understanding it is the key to understanding how you think, feel, and function as a human being. So we've established that neurons communicate through electrical signals. But how exactly does that work? How does a cell generate electricity? The answer lies in something called the action potential. And honestly, this is one of the most elegant systems in all of biology. Here's the setup. Every neuron has a membrane, a thin outer layer that separates the inside of the cell from the outside. This membrane is covered with tiny channels and pumps that control what goes in and out. Now, On either side of this membrane, you've got charged particles called ions. The main players here are sodium, which carries a positive charge, and potassium, which also carries a positive charge. There are also chloride ions, which are negative, and various proteins inside the cell that carry negative charges too. Here's the key point. When a neuron is just sitting there, not firing, it's in what we call the resting state. And in this resting state, there's an imbalance of charges across the membrane. The inside of the neuron is more negative than the outside. How much more negative? About negative 70 mil. This difference in charge is called the resting membrane potential. Why does this imbalance exist? It's maintained by a protein pump called the sodium-potassium pump. This pump constantly pushes 3 sodium ions out of the cell for every 2 potassium ions it brings in. Since more positive charges are being kicked out than brought in, the inside stays negative. Now, here's where the action happens. When a neuron receives enough stimulation from other neurons, something dramatic occurs. Voltage-gated sodium channels in the membrane suddenly open up. Remember all that sodium that's been building up outside the cell? It comes rushing in. Positive charge is flooding into the neuron. This causes a rapid change in the membrane potential. It goes from negative 70 millivolts all the way up to about positive 30 millivolts in less than a millisecond. This sudden reversal of charge is called deep, but it doesn't stop there. Almost immediately after the sodium channels open, TAE snap shut, and then voltage-gated potassium channels open up. Now potassium rushes out of the cell, taking positive charges with it. This brings the membrane potential back down, actually overshooting it slightly to about negative 80 millivolts before returning to the resting potential. This return to baseline is called repolarization, and that brief overshoot is called hyper. This entire sequence, from resting state to depolarization to repolarization, is the action potential, and it all happens in about 1 to 2 milliseconds. But here's the really clever part. This action potential doesn't just happen in one spot. It travels down the length of the axon like a wave. When one section of the membrane depolarizes, it triggers the section right next to it to depolarize too, and that section triggers the next one, and so on, all the way down the axon to the terminal. Think of it like dominoes falling. Once you knock over the first one, the rest follow automatically. Now, there's an important feature here called the all-or-nothing principle. Either a neuron fires completely, or it doesn't fire at all. There's no such thing as a partial action potential. It's like a gun. You either pull the trigger and the bullet fires with full force, or you don't pull it and nothing happens. There's no half pull that shoots the bullet halfway. For this to happen, the initial stimulation has to reach a certain threshold, typically around negative 55 millivolts. If the stimulation doesn't get the membrane potential up to that threshold, nothing happens. But once it crosses that line, bang, full action potential. Now, here's another fascinating aspect. After an action potential fires, there's a brief period where the neuron cannot fire again. This is called the refractory period. During the absolute refractory period, no matter how strong the stimulus, the neuron won't fire. This is because the sodium channels are inactivated and need time to reset. This typically lasts about 1 millisecond. Then there's the relative refractory period, where the neuron can fire again, but only if it receives a stronger than normal stimulus. This is because the membrane is hyperpolarized and needs an extra push to reach threshold. The refractory period is actually crucial for proper nervous system function. It ensures that action potentials travel in one direction, from the cell body toward the axon terminals, rather than bouncing back and forth. So we've got action potentials traveling down axons. But here's a question. What determines how fast these signals travel? The answer involves one of the most remarkable adaptations in the nervous system: myelin. Myelin is a fatty substance that wraps around many axons like insulation around a wire. This myelin sheath doesn't cover the entire axon continuously. Instead, it comes in segments with small gaps in between. These gaps are called nodes of Ranvier, named after the French anatomist Louis-Antoine Ranvier, who discovered them. Now, here's why myelin is so important. Remember how action potentials normally travel? They trigger the next section of membrane to depolarize, which triggers the next section, and so on. This process is called continuous conduction. But when an axon is wrapped in myelin, the action potential can't occur where the myelin is. Ion channels can't open through that fatty coating, so the electrical signal has to jump from one node of Ranvier to the next. This jump in propagation is called saltatory conduction, from the Latin word saltar, meaning to jump, and it dramatically increases the speed of signal transmission. How much faster? An unmyelinated axon might conduct signals at about 1 meter per second. A myelinated axon of the same diameter can conduct signals at over 100 meters per second. That's roughly 100 times faster. Think about why this matters. Your brain is about 1.5 meters away from your feet. If you're walking and your toe hits something, you need that signal to reach your brain quickly so you can respond. With myelinated axons, that signal arrives almost instantaneously. The myelin sheath is produced by specialized cells in the central nervous system, brain, and spinal cord. These cells are called A single oligodendrocyte can extend projections to myelinate segments of up to 50 different axons. In the peripheral nervous system, everything outside the brain and spinal cord, the job falls to Schwann cells. Unlike oligodendrocytes, each Schwann cell wraps around just one axon segment. This difference has real clinical implications. When myelin is damaged in the central nervous system, repair is difficult because oligodendrocytes don't regenerate well. But in the peripheral nervous system, Schwann cells can help repair damaged nerves, which is why peripheral nerve injuries often have better outcomes than central nervous system injuries. You've probably heard of multiple sclerosis. This is a disease where the immune system attacks myelin in the central nervous system. Without proper myelin, signals slow down or stop entirely. This leads to symptoms like muscle weakness, coordination problems, vision issues, and cognitive difficulties. The symptoms depend on which areas of the brain and spinal cord are affected, because that determines which signals are being disrupted. Another condition, Guillain-Barré syndrome, involves the immune system attacking myelin in the peripheral nervous system. This can cause muscle weakness and paralysis that typically starts in the legs and moves upward. Both of these conditions highlight just how critical myelin is for normal nervous system function. Now, not all axons are much smaller axons, particularly those carrying pain and temperature information, often lack myelin. This is why a painful stimulus might feel like it comes in waves. The initial sharp pain travels through faster myelinated fibers, while the duller, burning pain that follows comes through slower unmyelinated fibers. Alright, so an action potential has traveled down an axon and reached the terminal. Now what? This is where we get to one of the most amazing parts of neuroscience. This is the junction where one neuron communicates with the next cell, whether that's another neuron, a muscle cell, or a gland. Here's the thing. Neurons don't actually touch each other. There's a tiny gap between them, about 20 to 40 nanometers wide. To put that in perspective, a nanometer is 1/1000th of a meter. You'd need to line up about 25,000 of these gaps to equal the width of a single human hair. This gap is called the synaptic cleft, and the way signals cross this gap is through chemical messengers called neurotransmitters. Let me walk you through how this works, step by step. When an action potential arrives at the axon terminal, it triggers voltage-gated calcium channels to open. Calcium ions rush into the terminal from outside the cell. This influx of calcium is the signal for neurotransmitter release. Inside the terminal, there are small bubble-like structures called vesicles, and these vesicles are packed with neurotransmitter molecules. When calcium enters, it activates proteins called snare proteins. These proteins cause the vesicles to fuse with the cell membrane and dump their contents into the synaptic cleft. Now you've got neurotransmitter molecules floating in the gap between neurons. They drift across to the other side, the postsynaptic membrane, where they bind to specialized receptor proteins. What happens next depends on the type of receptor and the type of neurotransmitter. Some receptors are actually ion channels that open when the neurotransmitter binds, letting ions flow into or out of the postsynaptic cell. These are called ionotropic receptors, and they work fast. Other receptors, called metabotropic receptors, trigger a cascade of chemical reactions inside the cell when activated. These work more slowly, but can have longer-lasting effects. Now, here's something crucial. Neurotransmitters don't stay in the synapse forever. If they did, the signal would never stop. There are three main ways neurotransmitters get cleared out. First, they can be broken down by enzymes in the synaptic cleft. Acetylcholine, for example, is broken down by an enzyme called acetylcholine. Second, they can be taken back up into the presynaptic terminal through a process called reuptake. The neurotransmitter molecules are essentially recycled, packaged back into vesicles for future use. Third, they can simply drift away from the synapse and be removed through general diffusion. Many psychiatric medications work by targeting these clearance mechanisms. Selective serotonin reuptake inhibitors, commonly called SSRIs, and used to treat depression, block the reuptake of serotonin, keeping it in the synapse longer. Here's another important point. Synapses can be either excitatory or inhibitory. At an excitatory synapse, the neurotransmitter makes the postsynaptic neuron more likely to fire its own action potential. It does this by causing a slight depolarization, bringing the membrane potential closer to threshold. At an inhibitory synapse, the opposite happens. The neurotransmitter makes the postsynaptic neuron less likely to fire, usually by causing hyperpolarization or stabilizing the membrane potential. Any given neuron receives inputs from hundreds or even thousands of other neurons. Some of these inputs are excitatory, some are inhibitory. The neuron essentially adds up all these influences at the axon hillock, the region where the cell body meets the axon. If the sum of all inputs brings the membrane to threshold, the neuron fires. If not, it stays quiet. This integration of multiple inputs is how the nervous system processes complex information. We've talked about neurotransmitters in general. Now, let's get specific. Your body uses dozens of different neurotransmitters, each with its own functions and effects. Let's start with glutamate. This is the most common excitatory neurotransmitter in the brain, used in about 90% of excitatory settings. When glutamate binds to its receptors, it makes neurons more likely to fire. Glutamate is essential for learning and memory. It plays a huge role in synaptic plasticity, the ability of synapses to strengthen or weaken over time. This is thought to be the cellular basis of how we learn and remember things. But too much glutamate can be dangerous. Excessive glutamate release can overexcite neurons, to the point where they're damaged or killed. This is called excitotoxicity, and it plays a role in conditions like stroke and neurodegenerative diseases. Now let's talk about GABA, which stands for gamma-aminobutyric acid. This is the most common inhibitory neurotransmitter in the brain. When GABA binds to its receptors, it makes neurons less likely to fire. GABA is crucial for keeping neural activity balanced. Without enough GABA, neurons would fire uncontrollably. This is actually what happens during seizures, at least in many cases. There's either too little GABA activity or too much glutamate activity, and the balance tips toward excitation. Many medications that calm the nervous system work by enhancing GABA effects. Benzodiazepines, like Valium and Xanax, barbiturates, and alcohol increase GABA activity. This is why they make you feel relaxed and sleepy, and why combining them can be dangerous. Next up is acetylcholine. This was actually the first neurotransmitter ever discovered, identified in 1921 by Autonomic in a classic experiment involving frog hearts. Acetylcholine has many roles. In the peripheral nervous system, it's the neurotransmitter used at the neuromuscular junction, where motor neurons communicate with muscle fibers. Every time you move any muscle in your body, in the brain, acetylcholine is important for attention, memory, and arousal. The degeneration of acetylcholine-producing neurons is one of the hallmarks of Alzheimer's disease, which is why memory loss is such a prominent symptom. Now, let's talk about dopamine. This one gets a lot of attention in popular science, often called the pleasure chemical. But that's an oversimplification. Dopamine is involved in reward and motivation. When you achieve something or anticipate a reward, dopamine is released. But it's not just about pleasure. It's more about motivation and reinforcing behaviors that lead to rewards. Dopamine is also crucial for movement. The part of the brain that controls voluntary movement, the basal ganglia, relies heavily on dopamine. When dopamine-producing neurons in this area die off, you get Parkinson's disease. The tremors, stiffness, and difficulty initiating movements seen in Parkinson's are all related to dopamine deficiency. On the flip side, too much dopamine activity is associated with schizophrenia. Many antipsychotic medications work by blocking dopamine receptors. Serotonin is another neurotransmitter that's often discussed in the context of mood. About 95% of your body's serotonin is actually in your gut, where it helps regulate digestion. But the serotonin in your brain plays important roles in mood, sleep, appetite, and more. Low serotonin activity has been linked to depression and anxiety. The SSR, as we mentioned earlier, treat these conditions by keeping serotonin in synapses longer. Finally, let's mention norepinephrine, sometimes called noradrenaline. This neurotransmitter is part of your body's stress response system. It increases alertness, attention, and arousal. When you're in a stressful situation, norepinephrine helps mobilize your body and mind for action. It's also important for mood regulation, which is why some antidepressants target norepinephrine as well as serotonin. Now that we understand the basic machinery, let's look at the command center. The brain. The human brain weighs about 3 pounds. It's roughly the size of a cantaloupe, and it consumes about 20% of your body's energy, despite being only about 2% of your body weight. The brain is divided into 3 main parts: the cerebrum, the cerebellum, and the brainstem. Each has distinct structures and functions, but they all work together as an integrated system. Let's start with The cerebrum. This is the largest part of the brain, making up about 85% of its total weight. When you picture a brain with its wrinkled, walnut-like surface, you're mostly seeing the outer layer of the cerebrum is called the cerebrocortex. This is where most higher-level processing happens. It's only about 2 to 4 millimeters thick, but it contains around 16 billion neurons. The wrinkled appearance of the cortex exists for a reason. All those folds dramatically increase the surface area, allowing more neurons to be packed into the limited space inside your skull. If you smoothed out the cortex, it would cover roughly 2.5 square feet. The ridges between the folds are called gyri, and the grooves are called sulci. The deepest grooves are called fissures. The cerebrum is divided into two hemispheres, left and right, connected by a thick bundle of nerve fibers called the corpus callosum. This connection allows the two hemispheres to communicate and coordinate. You've probably heard about left brain versus right brain thinking. The reality is more nuanced than popular culture suggests, but there are some differences between the hemispheres. The left hemisphere generally controls the right side of the body, and vice versa. The left hemisphere is typically dominant for language in most people. The right hemisphere tends to be more involved in spatial reasoning and processing emotional content. But these generalizations have lots of exceptions, and both hemispheres contribute to most tasks. The two sides are constantly communicating through the corpus callosum. Each hemisphere is divided into 4 lobes, named after the skull bones that cover them. The frontal lobe is at the front of the brain, behind your forehead. It's the largest lobe and is involved in executive functions, like planning, decision. It also contains the primary motor cortex, which controls voluntary movement. The parietal lobe is toward the top and back of the brain. It processes sensory information related to touch, temperature, and body position. The primary somatosensory cortex is located here. The temporal lobe is on the sides of the brain, around your temples. It's crucial for hearing, language, comprehension, and memory. The auditory cortex is located here, as is a structure called the hippocampus, which is essential for forming new memories. The occipital lobe is at the very back of the brain. It's dedicated primarily to vision. The primary visual cortex processes information from your eyes, and surrounding areas interpret that information to recognize objects, faces, and colors. Now, beneath the cerebrocortex, you've got structures that make up the subcortical regions. These include the basal ganglia, which help control movement, and the limbic system, which is involved in emotion, memory, and motivation. The limbic system includes structures like the hippocampus, which we already mentioned for memory, and the amygdala, which plays a key role in processing emotions, especially fear. While the cerebrum handles higher-level thinking, other parts of the brain manage more fundamental functions. The brainstem connects the cerebrum to the spinal cord. It's located at the base of the brain and controls many vital functions that happen automatically without you having to think about them. The brainstem has 3 main parts: the midbrain, the pons, and the medulla oblongata. The medulla oblongata is the lowest part, connecting directly to the spinal cord. It controls some of the most critical functions for survival: heart rate, breathing, and blood pressure. Damage to the medulla is often fatal because these vital functions stop working. The medulla also contains reflex centers for vomiting, coughing, sneezing, and swallowing. These reflexes help protect your airway and digestive system. Above the medulla sits the pons. The word pons means bridge in Latin, and that's essentially what it is. It serves as a relay station, passing signals between the cerebrum and cerebellum. The pons also plays a role in regulating breathing and sleep. The midbrain is the top portion of the brainstem. It's involved in visual and auditory processing, eye movement, and motor control. The substantia nigra, a structure in the midbrain, produces dopamine, and is the area affected in Parkinson's disease. Running through the brainstem is a network called the reticular formation. This is a collection of nuclei that plays a crucial role in regulating consciousness and alertness. When you wake up in the morning, it's partly because your reticular formation has activated. 10 of the 12 cranial nerves originate from or pass through the brainstem, giving it connections to your face, eyes, ears, tongue, and many internal organs. Now let's talk about the cerebellum. The name means little brain, and it looks like a smaller, more tightly folded version of the cerebrum, located at the back of your head. Despite being only about 10% of the brain's volume, the cerebellum contains more than half of all the neurons in your brain, roughly 70 billion of them. The cerebellum's main job is coordinating movement. It doesn't initiate movements, but it fine-tunes them, making them smooth and accurate. When you reach for a glass of water and your hand goes directly to it without wobbling or overshooting, Thank your cerebellum. The cerebellum also helps with balance and posture. It receives information from your inner ear about your body's position in space and uses that to help you stay upright. People with cerebellar damage often have ataxia, a condition where movements are jerky, uncoordinated, and imprecise. They might have trouble walking in a straight line, their speech might be slurred, and they might have difficulty with tasks requiring fine motor control. Recent research has shown that the cerebellum is involved in more than just movement. It also seems to play roles in cognitive functions like language, attention, and even emotion. The cerebellum receives inputs from all over the brain and seems to help coordinate many different types of processing. Between the cerebrum and brainstem, you've got structures called idea. This includes the thalamus and hypothalamus, among other structures. The thalamus is like a relay station for sensory information. Almost all sensory information, except smell, passes through the thalamus on its way to the cerebral cortex. The thalamus helps filter and direct this information to the appropriate areas. The hypothalamus, despite being only about the size of an almond, controls many aspects of homeostasis. It regulates body temperature, hunger, thirst, sleep cycles, and hormonal release. It's the main link between the nervous system and the endocrine system. The brain gets most of the attention, but the spinal cord is equally essential. It's the main highway connecting your brain to the rest of your body. The spinal cord is a long, cylindrical bundle of nerve tissue that extends from the brainstem, specifically from the medulla oblongata down through the vertebral column. In adults, it's about 45 centimeters long and roughly the diameter of your little finger. Here's something interesting. The spinal cord doesn't extend all the way down to the bottom of your spine. It actually ends around the level of the first or second lumbar vertebra, roughly at your lower back. Below that point, the vertebral canal contains only nerve roots, not the cord itself. This is clinically important when doctors need to do a lumbar puncture, also called a spinal tap, to collect cerebrospinal fluid. They insert the needle below where the cord ends. This way, they can access the fluid-filled space without risking damage to the spinal cord itself. The spinal cord is protected by the vertebral column, your backbone. It's also surrounded by 3 layers of membranes called meninges, the same protective coverings that surround the brain. And floating within these membranes is cerebrospinal fluid, which cushions and nourishes the neural tissue. If you were to cut a cross-section of the spinal cord, you'd see 2 distinct regions. In the center, you've got gray matter, which contains cell bodies of neurons. Surrounding that is white matter, which consists mainly of myelinated axons. In the brain, this arrangement is reversed. The gray matter is on the outside, with white matter inside. But in the spinal cord, gray matter is central. The gray matter of the spinal cord is roughly ace-shaped or butterfly-shaped. The projections of this H are cold horns. The dorsal horns, pointing toward your back, receive sensory information coming in from the body. The ventral horns, pointing toward your front, contain motor neurons that send commands out to muscles. The white matter surrounding the gray matter is organized into columns called These columns contain tracts, which are bundles of axons traveling to or from the brain. Ascending tracts carry sensory information up to the brain. For example, when you touch something, the signal travels up through ascending tracts to reach the somatosensory cortex. Descending tracts carry motor commands from the brain down to the body. When you decide to move your hand, the signal travels down through descending tracts to reach the appropriate motor neurons. The spinal cord is divided into segments, and each segment gives rise to a pair of spinal nerves, 1 on each side. There are 31 pairs of spinal nerves, in total, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. Each spinal nerve is formed from 2 roots. The dorsal route carries sensory information into the cord. The ventral root carries motor commands out of the cord. These roots merge to form the spinal nerve, which then branches out to supply specific regions of the body. The dorsal root has a swelling called the dorsal root ganglion, which contains the cell bodies of sensory neurons. This is where the sensory information first enters the central nervous system. Each spinal nerve supplies sensation to a specific area of skin, called a derm. Doctors can use dermoto maps to figure out where spinal cord damage has occurred based on which areas have lost sensation. One of the most impressive things about the spinal cord is that it can coordinate responses without any input from the brain. These automatic responses are called reflexes. Reflexes are fast, involuntary responses to specific stimuli that are important for protection and survival, allowing your body to react to danger before you even have time to think about it. Think about what happens when you accidentally touch a hot stove. You pull your hand away almost instantly, before you consciously register the pain. By the time your brain realizes what happened, your hand is already safely away from the heat. This happens because the reflex arc, the neural pathway for the reflex, is processed in the spinal cord, not the brain. The signal doesn't have to travel all the way up to your brain and back down. It takes a shortcut. Let's walk through a typical spinal reflex, using the withdrawal reflex as an example. First, a sensory receptor in your skin detects a painful stimulus, like the hot stove. This receptor sends a signal along a sensory neuron toward the spinal cord. The sensory neuron enters the spinal cord through the dorsal root and synapses with neurons in the gray matter. Often, this includes an interneuron, a neuron that connects sensory and motor neurons. The interneuron, synapses, with a motor neuron in the ventral horn. This motor neuron sends a signal out through the ventral root to muscles in your arm. The muscles contract, pulling your hand away from the heat. This entire process happens in a fraction of a second, much faster than if the signal had to go to your brain first. But wait, there's more complexity. While you're pulling your hand away, other things need to happen simultaneously. The muscles on the opposite side of your arm need to relax so they don't fight the withdrawal. And if you need to maintain balance, your other limbs might need to adjust. This is achieved through additional neural connections in the spinal cord. Reciprocal inhibition ensures that when one muscle group contracts, the opposing group relaxes. Crossed extensor reflexes help maintain balance when you quickly shift weight. Another famous reflex is the patellar reflex, also known as the knee-jerk reflex. When a doctor taps just below your kneecap, your lower leg kicks forward. This reflex involves the stretch reflex pathway. The tap stretches the quadriceps muscle, stimulating sensory receptors called muscle spindles. These send signals to the spinal cord, which directly activate motor neurons, going back to the same muscle. The muscle contracts, causing the kick. This is actually the simplest type of reflex, a monosynaptic reflex, because there's only one synapse involved, directly connecting the sensory neuron to the motor neuron. Most reflexes involve more synapses and are called polysynaptic reflexes. Doctors test reflexes during neurological exams because they can reveal a lot about the health of the nervous system. Abnormal reflexes can indicate damage to specific spinal cord segments, peripheral nerves, or even the brain. Hyperactive reflexes, where the response is too strong, can suggest damage to upper motor neurons, those in the brain and descending tracts. Reduced or absent reflexes might indicate damage to lower motor neurons, those in the spinal cord or peripheral nerves. Everything we've discussed so far about the brain and spinal cord makes up the central nervous system. Now, let's venture outside into the peripheral nervous system. The peripheral nervous system, or PNS, includes all the neural tissue outside the brain and spinal cord. It's essentially the communication network that connects the central nervous system to every part of your body. The main structures of the PNS are nerves and ganglia. Nerves are bundles of axons wrapped in connective tissue, like cables containing many wires. Ganglia are clusters of neuron cell bodies located outside the central nervous system. Peripheral nerves are classified based on where they originate. Cranial nerves emerge directly from the brain, mainly from the brainstem. Spinal nerves emerge from the spinal cord. Let's start with the cranial nerves. There are 12 pairs of them, each designated by a Roman numeral and a name. Here's a quick rundown. The first two, the olfactory nerve and optic nerve, deal with smell and vision respectively. They're actually extensions of the brain itself. The third, fourth, and sixth nerves, oculomotor, trochlear, and abducens, all control eye movement. The fifth nerve, the trigeminal nerve, is the main sensory nerve for your face. It also controls the muscles you use for chewing. If you've ever had trigeminal neuralgia, an incredibly painful condition affecting this nerve, you know how important it is. The seventh nerve, the facial nerve, controls your facial expressions. Bell's palsy, a condition causing temporary facial paralysis on one side, involves this nerve. The eighth nerve, the vestibulocochlear nerve, handles hearing and balance. The ninth nerve, the glossopharyngeal nerve, is involved in taste, swallowing, and monitoring blood pressure. The 10th nerve, the vagus nerve, is a remarkable structure. Vagus means wander in Latin, and this nerve wanders all through your torso. It controls heart rate, digestion, and many other functions. It's the main component of the parasympathetic nervous system below the head. The 11th nerve, the accessory nerve, controls muscles in your neck and shoulders. The 12th nerve, the hypoglossal nerve, controls your tongue movements. There's a mnemonic to remember these. Oh, to touch and feel very good, velvet, heaven. Each first letter corresponds to a nerve in order. Now, for spinal nerves. There are 31 pairs, and they're named according to where they exit the vertebral column. The cervical region has 8 pairs, labeled C1 through C8. The thoracic region has 12 pairs. The lumbar region has 5 pairs. The sacral region has 5 pairs, and there's one coccygeal pair. Spinal nerves form networks called plexuses in certain regions. The cervical plexus, formed from C1 to C4, supplies the neck and diaphragm. The brachial plexus, from C5 to T1, supplies the arms. The lumbar plexus, from L1 to L4, supplies part of the lower abdomen and legs. The sacral plexus, from L4 to S3, supplies the buttocks, legs, and feet. Damage to these plexuses can cause weakness or paralysis in the regions they supply. Brachial plexus injuries, for example, can occur during difficult childbirth and may result in paralysis of the arm. The peripheral nervous system is functionally divided into 2 main parts: the somatic nervous system and the autonomic nervous system. Let's start with the somatic. The somatic nervous system is responsible for voluntary movements and for transmitting sensory information from the body to the central nervous system. The motor part of this system controls skeletal muscles, the muscles you consciously control when you decide to walk, write, or wave at someone. Your somatic nervous system carries those commands from your brain to your muscles. The pathway for voluntary movement starts in the motor cortex of the brain. Upper motor neurons send their axons down through the brainstem and into the spinal cord. In the spinal cord, they synapse with lower motor neurons in the ventral horn. These lower motor neurons send their axons out through the ventral roots of spinal nerves to reach skeletal muscles. At the junction between nerve and muscle, called the neuromuscular junction, the neurotransmitter acetylcholine is released, triggering muscle contraction. This is a direct pathway. There's only one synapse between the central nervous system and the muscle. This is different from the autonomic system, which we'll discuss later, where there are 2 neurons in the pathway. On the sensory side, the somatic system transmits information about touch, pressure, pain, temperature, and body position from sensory receptors throughout the body to the central nervous system. Sensory receptors in the skin, muscles, and joints detect stimuli and send signals along sensory neurons. These neurons enter the spinal cord through the dorsal route and either synapse locally for reflexes or send signals up to the brain for conscious perception. These sensory receptors themselves come in many types. Mechanoreceptors detect physical stimuli like pressure and vibration. Thermoreceptors detect temperature changes. Nociceptors detect potentially harmful stimuli that we perceive as pain. Proprioceptors detect body position and movement. Let's look at some specific mechanoreceptors in the skin. Meissner's corpuscles are located near the skin surface and detect light touch. They adapt quickly, meaning they respond when you first touch something but stop firing if the touch is maintained. This is why you stop noticing the feel of your clothes shortly after putting them on. Merkel discs, also near the skin surface, detect sustained pressure and fine details. They're important for reading braille or feeling textures. Pacinian corpuscles are deeper in the skin and detect vibration and deep pressure. Their structure, with multiple layers, like an onion, makes them particularly sensitive to rapid vibrations. Ruffini endings detect skin stretch and are important for sensing finger position when grasping objects. These different receptor types work together to give us our rich sense of touch. The density of receptors varies across the body, which is why some areas, like your fingertips, are much more sensitive than others, like your buck. Now, let's talk about the other major division of the peripheral nervous system, the autonomic nervous system, or ANS. The autonomic nervous system controls involuntary functions. Your heart beating, your lungs breathing, your digestive system processing food, your pupils adjusting to light. All of these happen automatically, without conscious effort on your part. The term autonomic comes from the Greek word for self-governing, and that's exactly what this system does. It governs itself, keeping your internal environment stable, even when external conditions change. The autonomic system is divided into 2 branches that generally have opposing effects: the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is often called the fight-or-flight system. It prepares your body for action when you face a threat or a stressful situation. Think about what happens when you're scared. Your heart rate increases, pumping more blood to your muscles. Your breathing gets faster, bringing in more oxygen. Your pupils dilate, letting in more light so you can see better. Blood is diverted away from your digestive system, which isn't a priority when you're running from danger, and toward your muscles. Glucose is released from storage into your bloodstream to provide quick energy. You might sweat, which helps cool your body during physical exertion. Your attention becomes focused and alert. All of these responses help you deal with the threat, whether by fighting or fleeing. The sympathetic nervous system originates from the thoracic and lumbar regions of the spinal cord, specifically T1 through L2. That's why it's sometimes called the thoracolumbar division. Preganglionic neurons have relatively short axons that synapse in ganglia located in chains running alongside the spinal column, called the sympathetic chain. From there, postganglionic neurons have longer axons that travel to target organs. The neurotransmitter used by postganglionic sympathetic neurons is mainly Neuron. This binds to adrenergic receptors on target tissue. The parasympathetic nervous system is the counterpart to the sympathetic system. It's often called the rest and digest system because it promotes functions that help you conserve energy and maintain normal body processes during calm times. Under parasympathetic influence, your heart rate slows down, your digestive system becomes more active, your pupils constrict, your airways narrow slightly since you don't need maximal airflow. Essentially, the parasympathetic system returns your body to baseline after the sympathetic system has activated it. The parasympathetic nervous system originates from two regions, the brain, where it's carried by certain cranial nerves in the sacral spinal cord. That's why it's sometimes called the craniosacral division. The most important parasympathetic nerve is the vagus nerve, cranial nerve X. It extends from the brainstem all the way down through the chest and abdomen, supplying parasympathetic input to the heart, lungs, and most of the digestive tract. In the parasympathetic system, preganglionic neurons have long axons that travel to ganglia located near or within target organs. From there, postganglionic neurons have short axons that innervate the target tissue directly. Both pre- and postganglionic parasympathetic neurons use acetylcholine as their neurotransmitter. This binds to muscarinic receptors on target tissue. The sympathetic and parasympathetic systems don't operate as simple on-off switches. Instead, most organs receive input from both systems, and the balance between them determines the organ's activity level. Take your heart, for example. Sympathetic stimulation speeds up your heart rate. Parasympathetic stimulation slows it down. At any given moment, both systems are active to some degree. The actual heart rate depends on which influence is stronger. At rest, parasympathetic tone, your vagus nerve keeps your heart rate lower than it would be otherwise. If we blocked vagus nerve activity with a drug, your heart rate would actually increase. During exercise or stress, sympathetic activity increases while parasympathetic activity decreases. Your heart rate raises because both the accelerator is pressed and the brake is released. This dual innervation allows for very fine control. It's much more precise than having just one system that turns on or off. Not all organs follow this pattern, though. Some tissues receive innervation from only one branch of the autonomic system. Blood vessels, in skeletal muscle, for instance, are mainly controlled by sympathetic neurons. Sympathetic activation causes these vessels to constrict. The parasympathetic system doesn't directly control them, though other factors, like local metabolic conditions, can cause the adrenal medulla, the inner part of the adrenal gland, is a special case. It's essentially a modified sympathetic ganglion. When the sympathetic system is activated, preganglionic sympathetic neurons stimulate the adrenal medulla to release epinephrine and norepinephrine directly into the bloodstream. These hormones, often called adrenaline and noradrenaline, circulate throughout the body and amplify sympathetic effects. This is why the sympathetic response can be so widespread and powerful. Let's look at how the two branches affect different organ systems. In the eyes, sympathetic activation dilates the pupils. Parasympathetic activation constricts them. In the lungs, sympathetic activation relaxes the airways, allowing more air in. Parasympathetic activation constricts them. In the heart, as we discussed, sympathetic activation speeds it up and increases the force of contraction. Parasympathetic activation does the opposite. In the digestive system, sympathetic activation slows down motility and secretion. Parasympathetic activation increases both, promoting digestion. In the bladder, sympathetic activation relaxes the bladder wall and contracts the internal sphincter, promoting urine storage. Parasympathetic activation contracts the bladder wall and relaxes the sphincter, promoting urination. Understanding these effects helps explain how stress affects the body. Chronic stress keeps the sympathetic system overactive, which can lead to high blood pressure, digestive problems, and other health issues. It also explains how certain drugs work. Beta blockers, used to treat high blood pressure and heart conditions, block the effects of sympathetic stimulation on the heart. There's actually a third division of the autonomic nervous system that often gets overlooked: the enteric nervous system. The enteric nervous system is embedded in the walls of your gastrointestinal tract. It's sometimes called the second brain, and for good reason. It contains roughly 500 million neurons, more than in your spinal cord. This system can operate independently of the brain and spinal cord. Even if all connections between your gut and central nervous system were severed, your enteric nervous system would still be able to coordinate digestion. The enteric nervous system controls the rhythmic contractions that move food through your digestive tract, called Parasympathetic. It regulates the secretion of digestive enzymes and hormones. It manages blood flow to support digestion. It even has roles in immune function and inflammation. The neurons of the enteric system are organized into two main networks, or plexuses. The myenteric plexus, also called Auerbach's plexus, lies between the muscle layers of the gut wall. It primarily controls gut motility. The submucosal plexus, also called Meissner's plexus, is closer to the gut lining. It mainly controls secretion and local blood flow. These plexuses contain all the neuron types you'd find in the central nervous system: sensory neurons, motor neurons, and They use dozens of different neurotransmitters, including serotonin, dopamine, and acetylcholine. The enteric nervous system doesn't work in complete isolation, though it communicates extensively with the central nervous system through the vagus nerve and through sympathetic connections. This gut-brain connection is a hot topic in current research. There's evidence that the gut and brain influenced each other in many ways. Stress and emotions can affect gut function, which is why you might get an upset stomach when anxious. Conversely, gut conditions might influence mood and mental health. The microbiome, the trillions of bacteria living in your gut, appears to interact with the enteric nervous system and may influence brain function through neural, hormonal, and immune pathways. Research is exploring connections between gut health and conditions like anxiety, depression, autism, and neurodegenerative diseases. It's a rapidly evolving field that's changing how we think about the relationship between our gut and our mind. Let's dive deeper into how you perceive the world. Everything you experience, every sight, sound, smell, taste, and touch, starts with sensory receptors. Sensory receptors are specialized structures that detect specific types of stimuli and convert them into neural signals. This conversion process is called transduction. Different receptors are tuned to different types of energy or chemicals. Photoreceptors respond to light. Mechanoreceptors respond to physical forces. Chemoreceptors respond to chemical substances. Thermoreceptors respond to temperature. Nociceptors respond to potentially damaging stimuli. Let's explore each type. Photoreceptors are found in the retina of the eye. There are two main types: rods and cones. Rods are extremely sensitive to light and are crucial for vision in dim conditions. You have about 120 million rods in each eye. They don't detect color, which is why things look gray in very low light. Cones are less sensitive, but allow for color vision. You have about 6 million cones, concentrated in the center of the retina. There are three types, each sensitive to different wavelengths of light: red, green, and blue. Your perception of all the colors you see comes from combining signals from these three cone types. Mechanoreceptors are diverse and found throughout the body. We've already discussed the ones in the skin, but mechanoreceptors also exist in other locations. In the inner ear, hair cells act as mechanoreceptors for hearing. Sound waves cause vibrations that bend hair-like projections on these cells, generating neural signals. Similar hair cells in the vestibular system detect head movement and position for balance. In muscles, muscle spindles detect stretch. They're important for reflexes and for your sense of body position, called proprioception. In tendons, Golgi tendon organs detect tension. They help prevent muscle damage by triggering relaxation when tension gets too high. In blood vessels, baroreceptors detect pressure. They help regulate blood pressure by signaling to the brain's chemoreceptors detect chemical substances. The obvious examples are tastes and smell. Taste receptors in taste buds respond to 5 basic tastes: sweet, salty, sour, bitter, and umami, which is a savory, meaty taste. Different receptors are tuned to different chemical compounds that represent these tastes. Olfactory receptors in the nose detect airborne chemicals. You have about 400 different types of olfactory receptors, allowing you to distinguish thousands of different odors. Chemoreceptors also exist internally. Receptors in the brainstem monitor carbon dioxide and pH levels in the blood, adjusting breathing rate accordingly. Receptors in the carotid bodies monitor oxygen levels. Thermoreceptors detect temperature changes. Cold receptors respond best to temperatures below skin temperature. Warm receptors respond best to temperatures above skin temperature but below painful heat levels. Interestingly, some substances can activate thermoreceptors directly. Menthol activates cold receptors, which is why mint feels cool. Capsaicin The compound in hot peppers activates heat and pain receptors, which is why spicy food feels hot. Nociceptors are pain receptors. Unlike other sensory receptors, they don't adapt over time. This makes sense. You want to continue feeling pain until you remove yourself from the damaging situation. Nociceptors respond to various stimuli: mechanical damage, extreme temperatures, and chemicals released by damaged tissue. The perception of pain is complex and influenced by many factors, including attention, emotion, and prior experience. Once sensory receptors detect a stimulus, the information must travel to the brain for conscious perception. Let's trace these pathways. For most sensations from the body, sensory neurons enter the spinal cord through the dorsal root. From there, the information ascends to the brain through organized pathways called ascending tracts. There are two main ascending systems for somatosensory information. The dorsal-columedial lemniscal pathway carries information about fine touch, vibration, and this is the high-definition channel, providing precise, detailed information. Sensory neurons enter the spinal cord and immediately ascend in the dorsal columns without synapse. They travel all the way up to the medulla, where they finally the signal then crosses to the opposite side of the brain and continues up through the brainstem to the from the thalamus. It goes to the somatosensory cortex. The spinothalamic tract carries information about pain and temperature and some crude touch. Sensory neurons enter the spinal cord and synapse. Immediately, the signal then crosses to the opposite side of the spinal cord and ascends in the spinothalamic tract to the thalamus, and then to the cortex. Notice that both pathways cross from one side to the other. This is why the right side of your brain processes sensations from the left side of your body and vice versa. The thalamus acts as a relay station for almost all sensory information going to the cortex. It filters and organizes information before passing it along. The primary somatosensory cortex, located in the parietal lobe, receives and processes touch information. Different parts of this cortex correspond to different body regions, creating a sensory map. This map is not proportional to the actual size of body parts. Areas with more sensory receptors, like the hands and lips, have larger representation in the cortex. This distorted body map is sometimes called the sensory Vision and hearing follow different pathways. Visual information from the retina travels through the optic nerve to the thalamus, specifically to a region called the lateral geniculate nucleus. From there, it goes to the primary visual cortex in the occipital lobe. Auditory information from the cochlea travels through the vestibulocochlear nerve to brainstem nuclei, then to the thalamus, specifically the medial genicular nucleus, and finally to the primary auditory cortex in the temporal lobe. Smell is unique because it's the only sense that doesn't pass through the thalamus on its way to the cortex. Olfactory information goes directly from the olfactory bulb to the olfactory cortex. It also has direct connections to the limbic system, which is why smells can evoke strong emotional memories. Sensation is only half the story. The other half is movement. Your nervous system must not only perceive the world but also act upon it. The motor system is responsible for planning, initiating, and executing movements. It's a hierarchy with multiple levels of control. At the top of this hierarchy is the motor cortex, located in the frontal lobe, just in front of the central sulcus. This region contains the primary motor cortex, premotor cortex, and supplementary motor area. The primary motor cortex directly controls voluntary movements. Like the somatosensory cortex, it has a topographic map of the body, called the motor home. Different regions control different body parts, with areas requiring fine motor control, like the hands and face, having larger representation. The premotor cortex and supplementary motor area are involved in planning and coordinating movements. They help sequence complex actions and prepare movements before they're executed. But the motor cortex doesn't work alone. It receives input from and sends output to many other structures. The basal ganglia, a group of structures deep within the cerebrum, play a crucial role in regulating movement. They don't initiate movements directly, but help select appropriate movements and inhibit unwanted ones. Diseases of the basal ganglia cause movement disorders. In Parkinson's disease, loss of dopamine input to the basal ganglia leads to difficulty initiating movement, tremors, and rigidity. In Huntington's disease, damage to the basal ganglia causes excessive, uncontrolled movements. The cerebellum, as we discussed earlier, fine-tunes movements and ensures they're smooth and coordinated. It receives feedback about body position and ongoing movements and adjusts motor commands accordingly. Signals from the motor cortex travel down to the spinal cord through descending tracts. The most important of these is the corticospinal tract, also called the pyramidal tract. This tract begins in the motor cortex. Axons descend through the brainstem, with most of them crossing to the opposite side, in the medulla, at a structure called the pyramidal dicosae. This crossing is why the left motor cortex controls the right side of the body. The corticospinal tract ends in the spinal cord, where upper motor neurons synapse with lower motor neurons in the ventral horn. Lower motor neurons then exit through the ventral root and travel to skeletal muscles. At the neuromuscular junction, lower motor neurons release acid, which binds to receptors on the muscle fiber and triggers contraction. Damage at different levels of this system produces different patterns of weakness. Upper motor neuron damage, affecting the brain or descending tracts, causes weakness with increased muscle tone and hyperactive reflexes. Lower motor neuron damage, affecting the spinal cord motor neurons or peripheral nerves, causes weakness with decreased muscle tone and reduced reflexes. Throughout this entire discussion, we've focused mainly on neurons. But neurons aren't the only cells in the nervous system. There's another category of cells that are equally important: glial cells, or glia. The word glia comes from the Greek word for glue. Early scientists thought these cells simply held neurons together, but we now know that glia are essential for nervous system function in many ways. In fact, glial cells actually outnumber neurons. Your brain contains roughly equal numbers of neurons and glia, but in total, there are more glial cells, because the ratio varies by region. There are several types of glial cells, each with distinct functions. Astrocytes are the most abundant glial cells in the brain. They're named for their star-like shape. Astrocytes perform many critical functions. They maintain the chemical environment around neurons, regulating levels of ions and neurotransmitters. They provide metabolic support, supplying neurons with nutrients and removing waste. They help form and maintain the blood-brain barrier, a selective barrier that protects the brain from harmful substances in the blood. Astrocytes also play active roles in synaptic function. They can release signaling molecules and influence how neurons communicate. This has led some researchers to speak of tripartite synapses, where astrocytes are active participants alongside the pre- and postsynaptic neurons. Oligodendrocytes are the cells that produce myelin in the central nervous system. A single oligodendrocyte can extend processes to myelinate segments of multiple axons. In the peripheral nervous system, Schwann cells perform the same function. However, each Schwann cell myelinates only one segment of one axon. When myelin is damaged, as in multiple sclerosis, signal conduction slows or stops. Oligodendrocytes have limited ability to regenerate, which is why MS damage is often permanent. Schwann cells are more capable of regeneration, which is why peripheral nerve injuries often heal better than central nervous system injuries. Microglia are the immune cells of the brain. They're constantly surveying their environment for signs of damage or infection. When they detect a problem, they can engulf and destroy pathogens and clear away debris from dead cells. Microglia are important for brain development too. During early life, they help prune unnecessary synapses, sculpting neural circuits into their mature form. However, in some disease states, microglia may become overactivated and contribute to inflammation and neuronal damage. This has been implicated in conditions ranging from Alzheimer's disease to chronic pain. Ependymal cells line the ventricles of the brain and the central canal of the spinal cord. They have tiny hair-like projections, called cilia, that help circulate cerebrospinal fluid. Some ependymal cells, in the choroid plexus, are responsible for producing cerebrospinal fluid. Satellite cells are found in the peripheral nervous system, surrounding neuron cell bodies in ganglia. They provide support and protection for these neurons, similar to what astrocytes do in the central nervous system. The nervous system is incredibly delicate. Your brain has the consistency of soft butter or firm gelatin. A sharp blow or sudden acceleration can cause serious damage. That's why the nervous system has multiple layers of protection. The brain is housed within the skull, and the spinal cord is surrounded by the vertebral column. These bony structures provide the first line of defense against mechanical injury. But bone alone isn't enough. Between the bone and the neural tissue are three membranes called meninges. The outermost layer is the dura mater, which means tough mother in Latin. It's a thick, fibrous membrane that lines the inside of the skull and vertebral canal. The dura is durable and protective. The middle layer is the arachnoid mater, named for its spiderweb-like appearance. Below the arachnoid is a space filled with cerebrospinal fluid called the subarachnoid space. The innermost layer is the pia mater, meaning gentle mother. It's a thin, delicate membrane that closely follows the surface of the brain and spinal cord, dipping into every groove and fold. Cerebrospinal fluid, or CSF, fills the subarachnoid space and the ventricles within the brain. It serves several important functions. CSF cushions the brain, reducing the impact of sudden movements. Because the brain floats in this fluid, it effectively weighs only about 25 grams, rather than its actual 1,400 grams. This buoyancy protects against damage from the brain's own weight. CSF also provides nutrients and removes waste products. It maintains the chemical environment around neurons, contributing to proper neural function. CSF is produced primarily in the choroid plexus, a network of blood vessels in the ventricles. About 500 milliliters are produced daily, though the total volume at any time is only about 150 milliliters. This means CSF is constantly being produced and reabsorbed, turning over several times a day. The blood-brain barrier is another crucial protective feature. This barrier consists of specialized cells lining the blood vessels in the brain, along with support from the cells of the blood-brain barrier are connected by tight junctions that prevent most substances from freely passing between them. Only specific molecules can cross, either through special transporters or by passing through the cell membranes themselves. This barrier protects the brain from toxins, pathogens, and fluctuations in blood chemistry. But it also presents challenges for drug delivery. Many medications that work well elsewhere, and the body can't reach the brain because they can't cross this barrier. The blood-brain barrier isn't present everywhere in the brain. Some regions, like the area postrema in the brainstem, have leaky barriers. This allows the brain to monitor blood chemistry directly, which is important for functions like detecting toxins and triggering One of the most remarkable features of the nervous system is its ability to change. This property is called neurotransmitters. For much of the 20th century, scientists believed the adult brain was essentially fixed, that its connections were established during development and couldn't change significantly afterward. We now know this isn't true. The brain can reorganize itself throughout life. New connections can form, existing connections can strengthen or weaken, and in some regions, new neurons can even be generated. Neuroplasticity is the basis of learning and memory. When you learn something new, you're literally changing the structure of your brain. At the synaptic level, learning involves changes in how strongly neurons communicate with each other. These changes are often summarized by Hebb's rule. Neurons that fire together wire together. When two neurons are active at the same time repeatedly, the connection between them strengthens. This is called long-term potentiation, or LTP. Conversely, when neurons don't fire together, their connections can weaken. This is long-term depression, or LTP. These processes involve actual physical changes at synapses. Existing synapses can grow larger and more effective. New synapses can form. Receptors can be added or removed from the postsynaptic membrane. Glutamate, the main excitatory neurotransmitter, plays a key role in these processes. The NMDA receptor, one type of glutamate receptor, is particularly important. It acts as a coincidence detector, opening only when both the presynaptic neuron releases glutamate and the postsynaptic neuron is already depleted. This property makes NMDA receptors crucial for associative learning, linking events that occur together in time. The hippocampus, located in the temporal lobe, is essential for forming new explicit memories. Damage to the hippocampus can prevent new memories from being formed, while leaving older memories relatively intact. Different brain regions handle different types of memory. The hippocampus and related structures handle declarative memories, facts and events you can consciously recall. The basoganglia and cerebellum are important for procedural memories, skills and habits that you demonstrate through performance. Neuroplasticity extends beyond synaptic changes. The brain can also reorganize at larger scales. After a stroke damages part of the brain, surrounding areas can sometimes take over some of the lost functions. This reorganization doesn't happen automatically, but requires active rehabilitation and practice. Musicians, who spend thousands of hours practicing, show enlarged representations of their instruments in their motor cortices. London taxi drivers, who must memorize thousands of streets, have larger hippocampi than average. These examples show how the brain physically adapts to the demands placed upon it. What you do with your brain shapes what your brain becomes. We spend about a third of our lives asleep. Sleep isn't just a passive state of unconsciousness. It's an active process critical for nervous system function. During sleep, your brain goes through several stages in repeated cycles. Non-REM sleep includes 3 stages, progressing from light sleep to deep sleep. Stage 1 is the transition from wake. Stage 2 is light sleep, where brain activity includes distinctive patterns called sleep spindles and cake. Stage 3 is deep sleep, characterized by slow brain waves, called delta waves. REM sleep, named for the rapid eye movements that occur, is when most vivid dreaming happens. During REM, your brain is highly active, almost as active as when you're awake. But your voluntary muscles are paralyzed, which prevents you from acting out your dreams. A typical night includes 4 to 6 cycles through these stages, with more deep sleep early in the night and more REM sleep later. Why do we need sleep? Research has identified several important functions. Sleep is critical for memory consolidation. During sleep, memories formed during the day are processed and stored more permanently. Studies show that learning followed by sleep leads to better retention than learning followed by an equivalent period of wakefulness. Sleep may also be when the brain clears out waste products. The glymphatic system, a waste clearance pathway that relies on cerebrospinal fluid flow, is most active during sleep. This might help prevent the buildup of toxic proteins associated with neurodegenerative diseases. Sleep is important for emotional regulation too. Sleep deprivation impairs the prefrontal cortex, reducing its ability to control emotional responses. People who are sleep deprived are more emotionally reactive and have poorer judgment. The regulation of sleep involves multiple brain regions. The hypothalamus contains neurons that promote both sleep and wakefulness. The brainstem regulates transitions between sleep stages. The thalamus relays sensory information to the cortex and changes its activity dramatically between sleep and waking. Neurotransmitters play important roles. Norepinephrine and histamine promote wakefulness. GABA promotes sleep. Adenosine, a byproduct of cellular metabolism, builds up during wakefulness and promotes sleep. Caffeine works by blocking adenosine receptors. Sleep disorders can have serious consequences. Insomnia affects the ability to fall or stay asleep. Sleep apnea involves repeated interruptions in breathing during sleep. Narcolepsy causes sudden sleep attacks and can include cataplexy, a sudden loss of muscle tone triggered by emotions. The nervous system doesn't spring into existence fully formed. It develops through a remarkable process that begins early in embryonic life and continues into young adulthood. During the 3rd week after conception, a flat sheet of cells called the ectoderm begins to form the nervous system. Part of the ectoderm thickens into the neural plate. The edges of this plate rise up and fold together, eventually fusing to form the neural tube. The neural tube is the foundation of the entire central nervous system. The front end develops into the brain, while the rest becomes the spinal cord. The hollow center of the tube becomes the ventricles of the brain and the central canal of the spinal cord. Neural crest cells, which form at the edges of the neural plate, migrate throughout the body and give rise to the peripheral nervous system, among other structures. As development proceeds, neurons are generated in massive numbers. New neurons are produced from stem cells lining the ventricles. These newborn neurons then migrate to their final positions, often traveling considerable distances guided by chemical signals and cellular scaffolds. Once neurons reach their destinations, they begin growing axons and then reaching out to make connections with other cells. A single neuron may extend axons over long distances, guided by molecular cues that attract or repel the growing tip. The initial connections are far from perfect. The developing brain produces more neurons and more synapses than it will ultimately need. This is followed by a period of pruning, where excess neurons die off and weak or unused synapses are eliminated. This pruning is activity dependent. Connections that are used become stronger and survive. Connections that aren't used weaken and are eliminated. This is why early experiences are so important for brain development. The patterns of neural activity during development help shape which circuits persist. Brain development doesn't stop at birth. In fact, some regions continue developing into the mid. The prefrontal cortex, involved in judgment, planning, and impulse control, is one of the last regions to fully mature. This helps explain some of the behavior patterns seen in adolescence. Myelination also continues 4 years after birth. Improved myelination increases processing speed and may underlie some of the cognitive gains seen during childhood and adolescence. Critical periods are windows during development when specific experiences must occur for normal development. For example, there's a critical period for visual development. If vision is impaired early in life, the visual cortex may not develop properly, even if vision is restored later. These critical periods reflect the brain's developmental programming. But plasticity continues throughout life, allowing for ongoing learning and adaptation. Understanding how the nervous system works helps us understand what happens when it goes wrong. Let's look at some common neurological and psychiatric conditions. Stroke occurs when blood supply to part of the brain is interrupted. Ischemic strokes, caused by a blocked blood vessel, are most common. Hemorrhagic strokes, caused by bleeding in the brain, are less common but often more severe. Without blood supply, brain tissue quickly dies. The effects depend on which area is affected. A stroke in the motor cortex might cause weakness or paralysis. A stroke and the language areas might cause difficulty speaking or understanding speech. Time is critical in stroke treatment. Medications or procedures that restore blood flow can limit damage if given quickly, usually within a few hours of symptom onset. Parkinson's disease involves the progressive loss of dopamine-producing neurons in the substantia nigra of the midbrain. The resulting dopamine deficiency in the basal ganglia causes tremors, rigidity, slow movement, and balance problems. Current treatments include medications that increase dopamine signaling, like levodopa, which is converted to dopamine in the brain. Deep brain stimulation, where electrodes deliver electrical impulses to specific brain areas, can also help control symptoms. Alzheimer's disease is the most common cause of dementia. It involves the accumulation of abnormal protein deposits in the brain, amyloid plaques outside neurons, and tau tangles inside them. The disease typically begins with memory problems, particularly difficulty forming new memories, which reflects early damage to the hippocampus. As it progresses, other cognitive functions decline. Current treatments have limited effectiveness. Research continues into therapies targeting the underlying disease processes. Multiple sclerosis is an autoimmune disease where the immune system attacks myelin in the central nervous system. This causes episodes of neurological dysfunction that may partly recover, at least early in the disease. Symptoms depend on where the damage occurs and can include vision problems, weakness, numbness, coordination difficulties, and cognitive issues. Epilepsy is a condition of recurrent seizures, which are episodes of abnormal electrical activity in the brain. Seizures can range from brief lapses in awareness to violent convulsions. Many people with epilepsy are well controlled with medication. Others may benefit from surgery, dietary approaches, or devices that stimulate the vagus nerve. Depression and anxiety are among the most common mental health conditions. While their exact causes are complex, they involve dysfunction in brain circuits regulating mood, particularly those using serotonin, norepinephrine, and dopamine. Treatments include medications that modulate these neurotransmitter systems, psychotherapy, and lifestyle interventions. In severe cases, treatments like electroconvulsive therapy or transcranial magnetic stimulation may be used. We've covered an enormous amount of ground in this crash course. But here's the truth. Despite everything we've learned, we're still in the early stages of understanding the brain. Consider these. We know there are roughly 86 billion neurons in the human brain, and we understand the basic principles of how they work. But mapping all the connections between them, the connectome, is a monumental task. Recent projects have mapped complete connectomes for simple organisms. The connectome of C. elegans, a tiny worm with just 302 neurons, took years to complete. Mapping the human brain at this level of detail remains a distant goal. Technology is advancing rapidly, though. New imaging techniques allow us to see brain activity with unprecedented resolution. Optogenetics allows researchers to control specific neurons with light. Brain-computer interfaces are allowing paralyzed individuals to control computers and robotic limbs with their thoughts. Artificial intelligence is being used to analyze brain data and build models of neural circuits. These computational approaches may help us understand how billions of neurons work together to produce thought, perception, and consciousness. Speaking of consciousness, that remains one of the deepest mysteries. How does the physical activity of neurons give rise to subjective experience? How do patterns of electrical and chemical activity become the feeling of seeing red, or the taste of chocolate, or the pain of a stubbed toe? This is the hard problem of consciousness, and despite much philosophical and scientific attention, we still don't have a satisfying answer. There are also practical challenges ahead. Neurodegenerative diseases like Alzheimer's and Parkinson's are becoming more prevalent as populations age. Finding effective treatments is a major priority. Mental health conditions affect hundreds of millions of people worldwide. Better understanding of their neural basis could lead to more effective therapies. And then there are questions about enhancement. As we understand more about how the brain works, we may develop ways to improve cognitive function, memory, or mood in healthy people. This raises profound ethical questions about human nature and fairness. The brain is the most complex object in the known universe. It's the source of everything you've ever thought, felt, or done. Understanding it is one of the greatest scientific challenges of our time. And here's the beautiful irony. We're using our brains to study themselves. The very organ we're trying to understand is the same organ doing the understanding. That journey of self-discovery continues. Every day, researchers around the world are adding new pieces to the puzzle. And maybe, just maybe, you've found this crash course inspiring enough to want to learn more. Because, here's the thing, your brain isn't just something to study. It's something to appreciate. Every memory you cherish, every skill you've learned, every person you love, all of it exists because of this 3-pound organ in your head. Your nervous system is you. And the more you understand it, the more you understand what it means to be human. Thanks for watching. If you found this valuable, share it with someone else who wants to understand how they work. And as always, stay curious. Before we completely wrap up, let's spend a bit more time on the special senses. These are the senses that involve dedicated organs and pathways: vision, hearing, taste, smell, and balance. Vision begins with light entering the eye through the cornea and pupil. The lens focuses this light onto the retina, the light-sensitive layer at the back of the eye. The retina contains those photoreceptors we discussed earlier: rods for dim light and peripheral vision, cones for color and detailed central vision. When light hits a photoreceptor, it triggers a chemical reaction in light-sensitive proteins called in rods. This protein is in cones. There are 3 different types of photopasins, each sensitive to different wavelengths. This chemical reaction leads to electrical changes in the photoreceptor. Interestingly, photoreceptors work backward from what you might expect. In darkness, they constantly release neurotransmitter. When light hits them, they release less. This decrease in neurotransmitter is the signal that light is present. The signal passes through several layers of cells in the retina. Bipolar cells receive input from photoreceptors and pass it to ganglion cells. The axons of ganglion cells form the optic nerve, which carries visual information out of the eye and toward the brain. At the optic chiasm, some fibers from each optic nerve cross to the opposite side. The result is that information from the left visual field of both eyes goes to the right side of the brain, and information from the right visual field goes to the left side. From the chiasm, fibers continue through the optic tract to the lateral geniculate nucleus of the thalamus, and then to the primary visual cortex in the occipital lobe. But that's just the beginning. From the primary visual cortex, information flows to many other areas for further processing. Some areas specialize in detecting motion, others specialize in recognizing objects. Still others are involved in recognizing faces. This distributed processing is why different types of brain damage can cause different types of visual problems. Someone might lose the ability to perceive motion while still being able to see stationary objects. Someone else might be able to see but unable to recognize faces, a condition called prosopagnosia. Color vision comes from comparing signals from the 3 cone types. If all 3 are equally stimulated, we see white. If only the long wavelength cones are strongly stimulated, we see red. The brain constructs our perception of color by comparing the relative activation of the 3 cone types. Color blindness occurs when 1 or more cone types are missing or abnormal. The most common form is red-green color blindness, which affects about 8% of men and less than 1% of women. Hearing begins when sound waves enter the ear canal and cause the eardrum to vibrate. These vibrations are transmitted through 3 tiny bones in the middle ear: the malleus, incus, and stapes, commonly called the hammer, anvil, and stirrup. The stapes connects to the oval window, an opening into the inner ear. When it vibrates, it creates pressure waves in the fluid-filled cochlea. The cochlea is shaped like a snail shell and contains the organ of Corti, where hearing actually happens. Hair cells in the organ of Corti have tiny projections called When pressure waves cause the basilar membrane to move, the stereocilia bend. The bending opens ion channels, leading to electrical signals that are transmitted to the brain through the cochlear nerve, part of the vestibulocochlear nerve. Different frequencies of sound cause maximum vibration at different positions along the cochlea. High frequencies are detected near the base, low frequencies near the apex. This tonotopic organization is maintained all the way to the auditory cortex. Hearing loss can occur for many reasons. Damage to the outer or middle ear causes conductive hearing loss. Damage to the hair cells or auditory nerve causes sensorineural hearing loss. Age-related hearing loss typically involves loss of high-frequency sensitivity. Balance is handled by the vestibular system, also in the inner ear. This system includes 3 semicircular canals, oriented in different planes, and 2 otolith organs. The semicircular canals detect rotational movement of the head. Each canal is filled with fluid. When your head turns, the fluid lags behind due to inertia. This fluid movement bends hair cells in the canal, generating signals about the rotation. The otolith organs, the utreco and sacula, detect linear, acceleration, and head tilt. They contain crystals called otoliths, resting on a gel layer over hair cells. Gravity and acceleration cause the otoliths to shift, bending the hair cells. Information from the vestibular system goes to the brainstem, cerebellum, and cortex. It's integrated with visual and proprioceptive information to give you a sense of your body's position and movement in space. Vestibular problems can cause dizziness, vertigo, and balance difficulties. Benign paroxysmal positional vertigo, one of the most common vestibular disorders, occurs when otoliths become dislodged and end up in the semicircular canals. Taste and smell are chemical senses, detecting molecules in food and in the air. Taste begins with taste buds, found mainly on the tongue but also in other parts of the mouth and throat. Each taste bud contains taste receptor cells that respond to chemicals dissolved in saliva. There are 5 basic tastes. Sweet detects sugars and other carbohydrates. Salty detects sodium and other minerals. Sour detects acids. Bitter detects many different compounds, some of which are toxic. Umami detects amino acids like glutamate, indicating protein. Different taste receptor cells are sensitive to different tastes. When a taste molecule binds to a receptor, it triggers a signal that's transmitted through cranial nerves to the brainstem, and then to the gustatory cortex in the insula. The old idea that different parts of the tongue detect different tastes has been disproven. All areas of the tongue can detect all basic tastes, though there may be slight regional differences in sensitivity. Smell is much more complex. You have about 400 different types of olfactory receptors, each capable of recognizing different chemical features. This allows you to distinguish thousands of different odors. Olfactory receptors are located in the olfactory epithelium, a small patch of tissue high in the nasal cavity. When airborne molecules dissolve in the mucus covering this tissue and bind to receptors, they trigger signals in olfactory neurons. These neurons project to the olfactory bulb, a structure at the base of the brain. From there, information goes to the olfactory cortex. Notably, this pathway doesn't pass through the thalamus, making smell unique among the senses. The olfactory system has strong connections to the limbic system, including the amygdala and hippocampus. This explains why smells can evoke powerful emotional memories, a phenomenon called the Proustian effect, after the writer Marcel Proust, who described how the smell of a Madeleine cookie triggered vivid childhood memories. Taste and smell work together to create flavor. What you perceive as the taste of food is actually a combination of taste, smell, texture, and temperature. When you have a cold and your nose is blocked, food seems bland because you're missing the smell component. Olfactory neurons are unusual in that they're replaced throughout life. New neurons are generated from stem cells in the olfactory epithelium and integrate into the existing circuitry. This ongoing neurogenesis may help maintain olfactory function despite constant exposure to the environment. Pain is one of our most important senses, yet it's also one of the most complex and least understood. Let's explore it more deeply. Pain serves a protective function. It alerts us to potential or actual tissue damage and motivates us to take action to prevent or minimize harm. But pain can also become chronic and debilitating, persisting long after any original injury has healed. Nociceptors are the sensory receptors for pain. Unlike other sensory receptors, they don't adapt. A painful stimulus continues to produce a pain signal as long as it's present. There are different types of nociceptors. Some respond to intense mechanical force. Others respond to extreme temperatures. Still others respond to chemicals released by damaged or inflamed tissue. Polymodal nociceptors respond to multiple stimulus types. When tissue is damaged, cells release various chemicals, including prostaglandins, bradykinin, and histamine. These substances sensitize nociceptors, lowering their threshold and increasing their response. This is why an injured area becomes more sensitive to pain. Pain signals travel to the spinal cord through 2 types of nerve fibers. A delta fibers are myelinated and conduct quickly, producing the initial sharp, well-localized pain. C fibers are unmyelinated and conduct slowly, producing the duller, longer-lasting ache that follows. In the spinal cord, pain signals synapse in the dorsal horn. From there, they ascend to the brain through several pathways, including the spinothalamic tract. Pain processing in the brain involves multiple regions. The somatosensory cortex handles these sensory discriminative aspects. Where is the pain, and how intense is it? The anterior cingulate cortex and insula handle the emotional affective aspects. How unpleasant is this experience? The prefrontal cortex contributes to cognitive evaluative aspects. What does this mean, and what should I do about it? Importantly, pain can be modulated at multiple levels. The nervous system has built-in mechanisms for turning pain down. Descending pathways from the brainstem can inhibit pain transmission in the spinal cord. These pathways use neurotransmitters, including serotonin, norepinephrine, and endogenous opioids, natural painkillers, produced by the body. This explains phenomena like stress-induced analgesia, where pain is suppressed during emergencies, and why activities like exercise can reduce chronic pain. Opioid medications work by mimicking endogenous opioids, binding to opioid receptors in the brain and spinal cord to reduce pain signaling. The gate control theory suggests that known painful stimulation can close a gate in the spinal cord that reduces pain transmission, which may explain why rubbing an injury can help it feel better. We've explored the nervous system piece by piece, but in reality, all these components work together as an integrated whole. Let's consider how this integration produces unified experience and behavior. At any moment, your brain is receiving information from millions of sensory receptors. It's simultaneously maintaining your body position, regulating your internal organs, accessing memories, processing emotions, and potentially planning future actions. How does the brain coordinate all of this? Part of the answer involves parallel processing. Different brain regions handle different tasks simultaneously. Your visual cortex processes what you see, while your auditory cortex processes what you hear. Neither has to wait for the other. But this raises a question. How are these parallel streams integrated into a unified experience? When you watch a movie, you don't perceive the images and sounds as separate streams. They're bound together into a coherent whole. This binding problem is a major focus of neuroscience research. One theory suggests that synchronous neural oscillations help bind information from different areas. When neurons in different regions fire in synchrony, their information may be experienced as related. The concept of attention is crucial for integration. At any moment, you're aware of only a tiny fraction of all the sensory information your brain is receiving. Attention acts like a spotlight, selecting what reaches conscious awareness. Attention involves networks spanning the frontal and parietal lobes. These networks can be directed voluntarily, like when you focus on a particular conversation at a noisy party. They can also be captured automatically by salient stimuli, like a sudden loud noise. And then there's consciousness itself, the subjective experience of being aware. Despite decades of research, consciousness remains one of the greatest mysteries in science. We know that consciousness can be altered. Anesthesia eliminates it. Sleep changes it. Certain brain injuries can produce altered states. Studying these conditions has given us clues about what neural activity is necessary for consciousness. Some theories propose that consciousness arises from information integration across widespread brain networks. Others suggest it's related to specific patterns of neural activity or specific brain regions. What we do know is that consciousness isn't localized to one spot. It seems to emerge from the coordinated activity of many brain areas. The thalamus, cortex, and connections between them all appear to be important. Some researchers distinguish between levels of consciousness, how awake or aware you are, and contents of consciousness, what you're experiencing. These may involve different neural mechanisms. And here we are. 30 parts later, we've traveled from individual ions, passing through membrane channels, to the grand mystery of consciousness. We've explored neurons and synapses, brains and spinal cords, senses and movements, health and disease. But I want to leave you with something more than facts and figures. Your nervous system is a miracle of over hundreds of millions of years. Simple networks of nerve cells gradually became the staggeringly complex brain you carry in your head today. Every thought you think, every decision you make, every love you feel, every fear you overcome. It's all the product of electrochemical activity in this 3-pound organ. And here's what's really amazing. Your brain isn't just passively recording the world. It's actively constructing your experience of reality. The colors you see, the sounds you hear, the sensations you feel. None of these exist objectively in the outside world, the way you experience them. They're created by your brain based on patterns of neural activity. Your memories aren't perfect recordings either. Each time you remember something, your brain reconstructs it, and it can change a little each time. Your sense of self, your continuous identity over time, is also construction, a story your brain tells itself. This doesn't make these experiences less real or meaningful. It just means that the relationship between you and your brain is more intimate than you might have thought. You don't just have a brain. In a very real sense, you are your brain. Understanding this is empowering. If your brain constructed the patterns of thought and behavior that cause you problems, it can also change them. Neuroplasticity means you're not stuck with the brain you have. Through learning, practice, therapy, and sometimes medication, you can reshape your neural circuitry. It's also humbling. The more we learn about the brain, the more we realize how much automatic processing happens below the level of consciousness. Many of the decisions you think you're making consciously are actually being made by brain processes you're not aware of. But perhaps most importantly, understanding the nervous system should fill you with wonder. The fact that organized matter can become aware of itself, that evolution can produce consciousness from unconscious chemistry, that you can look up at the stars and wonder about your place in the universe. These are profound mysteries. Scientists are making progress, but we're far from fully understanding how the brain works. That means if you're interested in this field, there's a lifetime of discovery ahead. Whether you're a student preparing for exams, a healthcare professional deepening your knowledge, or simply a curious person who wanted to understand yourself better, I hope this crash course has been valuable. Your nervous system is your most precious possession. Take care of Italiano. Sleep well. Manage stress. Stay physically and mentally active. Protect your head, and feed your brain with good nutrition and rich experiences. And keep learning, because every time you learn something new, you're literally changing your brain. You're adding to the remarkable structure that makes you who you are. Thanks for spending this time with me. Until next time, take care of that amazing nervous system of yours.